So imagine you're on a rotation in the ER, right? And a patient comes in complaining of just intense muscle cramps.
Okay, sounds fairly standard so far.
Right, but they just went for a really light jog. Nothing crazy, but their
Yeah.
muscles are completely seizing up. And the really alarming part, they mentioned their urine has turned the color of dark cola.
Oh, wow. Yeah, that's a massive red flag.
Exactly. I mean, you might instantly start thinking about, rhabdomyolysis from a crush injury
Right.
or some severe trauma, but there's no trauma here at all.
Right, no crush injury.
None. The culprit isn't a muscle tear or a broken bone. It's actually a single, invisible, missing enzyme.
Yeah, the machinery of the muscle cell is essentially tearing itself apart just because it can't access its own fuel supply.
It's wild to think about.
It really is. I mean, we're so used to seeing medicine as gross anatomy-like, a broken bone on an X-ray or a visible tumor on a CT scan.
Right, the stuff you can physically see.
Exactly. But the real operating system of the human body runs entirely in the background. It's this microscopic, highly volatile environment where millions of lines of biochemical code are executing simultaneously.
Which is exactly why we're pulling you, our dedicated medical learner, into this deep dive today.
Welcome to the show. I'm your host, and we are going to get into the weeds of this stuff.
And I'm the resident expert here to help decode it all.
Today, we're looking at the biochemical software that keeps your patients alive and, well, what happens when that software encounters a fatal error?
Our guide for this deep dive is the material from Rapid Review Biochemistry.
But don't worry, we're not here to read you a textbook or, help you memorize endless pathways.
No, definitely not.
Our mission is to decode the logic of the body. We want to figure out the why and the how behind things like cellular pH, enzyme kinetics, and systemic metabolic collapse.
And to understand the collapse, you first really have to understand the baseline architecture. I mean, any operating system needs raw materials, right? Right. And for the human body, the most critical fuel source is carbohydrates, specifically glucose.
Exactly. The brain demands it almost exclusively under normal, non-starting conditions. But since we obviously don't eat continuously, we need a highly efficient way to warehouse that glucose.
Which brings us to how we store it. The sources emphasize how animals store glucose as glycogen, while plants store it as cellulose.
Both are just massive chains of glucose molecules, essentially.
Yeah, but looking at the biochemistry, the functional difference all comes down to the shape of the chemical bonds, right? Like cellulose uses these beta-1-go-4 glycosidic linkages.
Which stack into these very rigid fibrous sheets.
Right, and we humans lack the enzymes to break that beta bond. Which is why eating celery gives us, dietary fiber, but virtually zero caloric energy.
Right, we just can't unpack it. And contrast that with our own glycogen, which uses alpha-1-vol linkages. But the key feature isn't just the alpha bond, it's the branching.
The branch?
Yeah. Glycogen has these alpha-1, 4, and 6 branches every 8 to 10 residues, which creates this massive, highly branched tree structure.
Okay, so why does the branching matter so much? I mean, wouldn't a straight chain just be easier to pack into a cell?
Well, a straight chain would pack really tightly, like an unspooled ball of yarn, sure, but it would be an absolute nightmare to unpack in an emergency.
Oh, I see. Because of the ends.
Exactly. Because glycogen is highly branched, it has countless non-reducing ends. So when your blood sugar drops and the body secretes glucagon, the enzyme glycogen phosphorylase can attack thousands of those ends all at the same time.
Wow, okay, so it just snaps off glucose molecules like leaves off a tree in a hurricane.
That's a perfect way to put it. It's this brilliant evolutionary adaptation for rapid energy mobilization.
So carbs are the fuel, stored for rapid deployment. But then we have proteins, which seem to be the actual machines executing the commands, right?
Oh, absolutely. They do a bit of everything. I mean, collagen provides structural support. The sodium-potassium ATPase pump transports ions to maintain cell voltage.
And hemoglobin acts as an oxygen shuttle.
Right. But their most critical role in biochemistry is acting as catalysts. The vast majority of enzymes are proteins.
And the defining characteristic of proteins is that their function is entirely dependent on their three-dimensional shape.
Yes. That folded structure is held together by very delicate non-covalent bonds. Hydrogen bonds, ionic bonds, hydrophobic interactions.
Which honestly brings up a massive vulnerability. The environment required to keep those delicate bonds intact is incredibly narrow.
Very near.
Like the blood pH has to stay strictly between 7.35 and 7.45. I've always memorized the Henderson-Hasselbalch equation and the normal lab values, but practically, how is the body handling an influx of acid without the pH just plummeting?
So it relies heavily on the carbon dioxide and bicarbonate buffer system. It's a dynamic equilibrium. The body basically maintains a mixture of a weak acid and its conjugate base.
Okay, so when an influx of strong acid enters the system,
The bicarbonate ions intercept those free protons. They turn into carbonic acid, which then dissociates into water and carbon dioxide.
and then the lungs immediately throw off that extra carbon dioxide, right?
Exactly. It's a continuous integrated effort. The kidneys manage the bicarbonate side, and the lungs manage the volatile carbon dioxide side.
I'm trying to visualize this. It sounds kind of like the cooling system in a massive server room.
Ooh, I like that. Go on.
So if tissue hypoxia forces cells into anaerobic glycolysis, which generates lactic acid, or if, say, uncontrolled diabetes floods the blood with keto acids,
Right.
it's like a sudden massive heat wave in that server room. The buffer system has to aggressively absorb that excess heat, or in this case the protons, to prevent the ambient temperature from spiking.
That is a brilliant way to frame it, because if that cooling system fails and the patient enters metabolic acidosis, the heat essentially melts the
It
servers.
destroys the proteins.
Yeah, low intracellular pH, heavy metals, or literal heat will disrupt those stabilizing bonds inside the proteins. The polypeptide chains just unfold, exposing their hydrophobic cores to the watery environment of the cell.
And hydrophobic things hate water. So they're going to clump together to hide from it?
Precisely. They aggregate into these completely insoluble, non-functional masses. The protein is denatured. The sources actually give a fascinating clinical correlation for this.
Oh, G6PD deficiency.
Yes, glucose 6-phosphate dehydrogenase deficiency. In these patients, the red blood cells cannot neutralize oxidative stress. The peroxide just builds up and causes oxidative damage to the hemoglobin proteins.
So the hemoglobin denatures, folds inside out, and clumps up.
Yep.
And when you look at a blood smear under a microscope, you can actually see these dense, insoluble aggregates attached to the red blood cell membrane, right? The Heinz bodies.
The Heinz bodies, exactly. You are literally looking at the crashed software of the cell, and the spleen will eventually recognize those damaged cells and bite those Heinz bodies right out, which leads to hemolytic anemia.
Wow. It all traces back to protein stability.
It really does.
So if these proteins are so incredibly fragile, and a sudden drop in pH or oxidative stress can denature them, how do the proteins doing the heaviest lifting, our enzymes, manage to survive and drive thousands of chemical reactions without changing the final chemical equilibrium?
Well, they do it by manipulating activation energy. Think of a chemical reaction like pushing a boulder over a hill.
Okay.
The start and end points, like the equilibrium, they don't change. But an enzyme essentially tunnels straight through the hill. It provides an alternative pathway that requires significantly less initial energy to get the reaction going.
Makes sense. And the texts outline different ways these enzymes behave, right? Like we have Michaelis-Menten kinetics, which produce a hyperbolic curve.
All right, the classic curve.
As you add more substrate, the reaction speeds up, but eventually it flattens out as you hit Vmax, the maximum velocity. I always picture Vmax like a toll booth on a busy highway.
Okay, a toll booth.
Yeah, like it doesn't matter if there are 10 cars or 10,000 cars backed up. The toll booth operator can still only process one car every 5 seconds. The system is just saturated.
That is a perfect parallel. And in that analogy, the Michaelis constant represents the affinity of the enzyme for its substrate.
Okay, so how does Kalman fit into the toll booth?
It's essentially a measure of how efficiently the cars can find their way into the toll booth lane. A low comma means high affinity, so the enzyme grabs its substrate very easily, even at very low concentrations.
Ah, got it. So I follow Michaelis-Menten, but the sources also describe cooperativity kinetics, which produce a sigmoidal curve, like an S shape.
Yes, the S curve.
Why does it look like an S? What is physically happening to the enzyme to cause that?
So cooperativity usually happens in complex proteins with multiple binding subunits, like hemoglobin. At first, the enzyme is stubborn. It's in a tense state.
Tense state, okay.
Yeah, so it takes a lot of substrate to get the first molecule to bind. That's the flat bottom of the S. But once that first molecule forces its way in, it physically alters the three-dimensional conformation of the entire protein complex.
Oh, so it shifts shape.
Exactly. It forces the other subunits into a relaxed state. Suddenly, the second, third, and fourth binding sites are wide open and highly receptive.
So binding the first molecule makes binding the next one exponentially easier.
Yes.
The reaction rate shoots straight up, which is the middle of the S, before eventually plateauing when all sites are full. That is an incredible mechanism.
It's super elegant.
And these enzymes don't always do this alone, right? They rely heavily on coenzymes. I noticed a massive theme in the material. So many of these metabolic cofactors are directly derived from dietary vitamins.
Oh, yeah. It's the biological reason why vitamin deficiencies cause such systemic havoc. Take NAD+, for example. It's absolutely mandatory for oxidation reduction reactions in glycolysis.
And NAD plus is derived from niacin, right?
Right.
Yep. Then you have pyridoxal phosphate, which handles the transamination reactions needed to process amino acids. That comes from pyridoxine. Tetrahydrofolate handles one-carbon transfers and is derived from folic acid.
So basically, if you lack the vitamin, you lack the coenzyme.
Exactly. The enzyme sits there empty-handed, and the entire metabolic pathway just halts.
Let's pivot slightly and talk about what happens when tissues undergo necrosis, and these cellular machines are actually dumped into the bloodstream.
Diagnostic enzymology.
Yes. The section on diagnostic enzymology states that normal blood plasma has very few active enzymes in it. I've seen patients' lab results with elevated AST or lipase, but why are those enzymes in the blood in the first place? I thought they belonged strictly inside the hepatocytes or pancreatic cells.
Well, they do. Enzymes appearing in the serum are literally the biochemical debris of cell death.
Oh, wow.
Yeah, when a tissue becomes ischemic or inflamed, the cell membrane loses its integrity. It ruptures, and the intracellular contents just spill out into the systemic circulation.
And because different organs synthesize different specific enzymes.
We can read that debris field to pinpoint exactly which organ is dying.
So it's essentially a highly specific cellular SOS signal. If I see a spike in amylase lipase, I know the pancreas is inflamed. If I see AST aspartate aminotransferase spiking, I should be looking for a myocardial infarction or liver damage.
Exactly. Let's take that a step further with creatine kinase, or CK. This is where isoenzymes become critical diagnostic tools.
Isoenzymes.
Yeah, they're slightly different structural forms of the same enzyme. So CKMB is concentrated in the heart muscle. If it spikes, you suspect a heart attack.
Okay, what about the other forms?
Well, the CKMM is concentrated in skeletal muscle. So if a child presents with profound muscle weakness and massively elevated CKMM, you're suspecting Duchenne muscular dystrophy. We are literally tracing the biochemical breadcrumbs back to the failing organ.
And we have to remember the ultimate goal of all these cellular enzymes, right? The grand unifying currency of the body, generating ATP. The sources highlight that ATP's massive negative delta G is what makes life possible.
Right, because thermodynamics dictate that reactions only happen spontaneously if they release free energy. If they have a negative delta G.
But the body has thousands of reactions that require an input of energy. They'd never happen on their own.
Exactly. So the body couples them. It takes a reaction that requires energy and chains it to the breaking of an ATP bond, which releases an enormous amount of energy.
The net result is negative, and the biological work gets done.
Nailed it.
This logic brings us to an incredible crossroad. We know how enzymes mobilize energy, and we know they can spill into the blood when cells die. But what happens when the enzymes required to retrieve stored energy are genetically absent from birth?
The glycogen storage diseases, the GSDs.
Yes. Let's dive into those.
These disorders illustrate the concept of a metabolic bottleneck perfectly. When a single enzyme in a multi-step pathway is defective, the flow of metabolites just hits a brick wall.
The product downstream vanishes, and the substrate upstream accumulates to toxic levels.
Exactly. Von Guericke's disease is the classic example in the text. It's an autosomal recessive disorder where the patient is missing glucose 6-phosphatase.
So the liver successfully breaks down glycogen all the way to glucose 6-phosphate, but it can't perform the very final step. It can't cleave off that phosphate to release free glucose into the blood.
Right, and the clinical presentation is entirely predictable if you know the pathway. Because the liver can't release glucose, the patient suffers profound, severe fasting hypoglycemia.
And what happens to all that trapped glucose 6-phosphate?
Because it's trapped inside the liver cells, it acts as an osmotic drag, pulling in water, while also stimulating further glycogen synthesis.
Oh, man.
Yeah. The liver and kidneys become massively engorged with trapped glycogen, causing hepatourinomegaly.
That sounds awful. Compare that to Pom's disease. Here, the defect is in a lysosomal enzyme, alpha-1,4-glucosidase.
Right, POMS.
The glycogen just relentlessly builds up inside the lysosomes, primarily in the heart tissue. The infant form presents with profound cardiomegaly and leads to devastatingly early death.
It's tragic.
But the one that really caught my eye was McArdle's disease, which brings us back to the patient in the ER from our hook at the very beginning of the deep dive.
Yes. McArdle's is a deficiency of muscle glycogen phosphorylase.
And the presentation is completely different from von Guericke's. These patients have a totally normal fasting blood sugar. But the second they do strenuous exercise,
They get agonizing muscle cramps, fatigue, and myoglobinuria.
which explains the dark, cola-colored urine, their muscles are literally breaking down. Looking at these three diseases, there's an amazing pattern here. It seems like liver enzyme defects cause systemic fasting hypoglycemia because the liver acts as the body's altruistic food bank.
Right.
But muscle enzyme defects only cause local cramping during exertion because muscles are entirely selfish. They hoard glycogen only for their own contraction.
That is a phenomenal observation. I mean, the liver's biological mandate is maintaining systemic blood glucose for the brain. When its machinery breaks, the whole organism starves.
But the muscle is different.
Completely different. Skeletal muscle's mandate is mechanical work. It lacks glucose 6-phosphatase entirely, even in healthy individuals, because it never intends to share its glucose.
So when the muscle's internal retrieval pathway breaks, only the muscle suffers and only under load.
Exactly. And these bottlenecks aren't just limited to carbohydrates. The sources detail terrifying bottlenecks in lipid and amino acid metabolism as well. Take MCAD deficiency, medium-chain acyl-CoA dehydrogenase deficiency.
Okay, MCAD. What's the defect there?
It's a defect in the first step of beta oxidation for medium-chain fatty acids. When fasting, the body normally transitions to burning fat to spare glucose. But in MCAD deficiency, that lipid pathway hits a wall.
I noticed the text specifies this causes non-ketotic hypoglycemia. Now, I understand the hypoglycemia. If the body can't burn fat, every tissue starts aggressively consuming the remaining blood glucose, draining the tank fast.
Right.
But why is it non-ketotic? Wouldn't a starving body normally pump out ketones?
Normally, yes. But think about how ketones are synthesized. The liver produces ketone bodies directly from the massive amounts of acetyl-CoA generated during the beta oxidation of fatty acids.
Oh. And in MCAD deficiency, beta oxidation is broken.
Exactly. You have no fatty acid breakdown, which means you have no acetyl-CoA surplus. Therefore, you cannot synthesize ketones. The brain is deprived of both glucose and its backup fuel, leading to profound lethargy, vomiting, and potential coma.
That is terrifying. And the text mentions a fascinating toxicological mimic of this pathway called Jamaican vomiting sickness. It happens when someone eats the unripe fruit of the ackee tree.
Right, the ackee fruit.
The fruit contains a toxin called hypoglycin, and hypoglycin specifically inhibits acyl-CoA dehydrogenases.
It acts as an artificial bottleneck. Whether the enzyme is missing due to a mutated gene or paralyzed by a tropical toxin, the biochemical fallout is identical.
The flow stops, the body scrambles for alternative fuels, and catastrophic hypoglycemia ensues.
Exactly. And we see similar pathological buildups in sphingolipidoses. Diseases like Tay-Sachs, Gaucher's, and Niemann-Pick all stem from defects in lysosomal enzymes responsible for degrading complex lipids.
The sphingolipids accumulate to toxic levels, typically devastating the central nervous system.
Right. Or phenylketonuria, PKU. A deficiency in phenylalanine hydroxylase means the patient cannot convert the amino acid phenylalanine into tyrosine.
So phenylalanine builds up, gets shunted into toxic secondary metabolites, and causes severe intellectual disability if the patient doesn't immediately adhere to a strict lifelong diet.
It's all about the bottlenecks.
So if we pull back the lens, the body has all these distinct pathways. Glycolysis, beta oxidation, glycogen synthesis. How does the body coordinate this massive chaotic factory? Like, how does it know when to store fuel versus when to burn it?
It relies on chemical broadcasting. Hormones basically act as the grand conductors of the metabolic orchestra.
The conductors.
Yeah. The major players here are insulin, glucagon, and epinephrine. And they exert their power primarily by adding or removing phosphate groups from the critical rate-limiting enzymes in those pathways we just discussed.
So insulin is the ultimate hormone of plenty, right? The text notes it's synthesized by the pancreatic beta cells as proinsulin, which is then cleaved into a C-peptide and the active insulin molecule.
Correct.
When you eat a heavy meal, insulin floods the blood. It exerts a hypoglycemic effect, meaning its goal is to pull glucose out of the blood and into the tissues. It upregulates glycogen synthesis, it drives lipid synthesis, it tells the cellular factory we have an abundance of materials, build and store.
And on the opposite side of that metabolic seesaw, you have glucagon and epinephrine. These are the hormones of mobilization and stress.
They recognize that blood glucose is dropping and they exert a hyperglycemic effect to rescue it. I want to spend a second on how they actually do this, because the mechanism in the text involves G-protein coupled receptors, and it is a really beautiful piece of machinery. How does a hormone sitting on the outside of a cell membrane change the behavior of an enzyme deep inside the cytoplasm.
Okay, so it happens via a signal transduction cascade. Glucagon or epinephrine binds to a specific receptor on the cell surface.
GPCR.
Yes. That receptor undergoes a conformational change that activates a stimulatory G protein, or Gs, attached to its inner surface.
And then?
That activated G protein slides over and turns on an enzyme called adenylate cyclase.
And adenylate cyclase acts like an amplifier, right? It takes ATP and rapidly converts it into cyclic AMP or CAMP, the secondary messenger.
Exactly. The CAMP levels inside the cell skyrocket. That CAMP then activates protein kinase A. And remember, kinases are enzymes that add phosphate groups to other proteins.
So protein kinase A goes on a phosphorylation spree.
It really does. It attaches phosphates to the enzymes responsible for glycogen breakdown and lipolysis, instantly turning them on, while simultaneously turning off the enzymes responsible for storage.
It's an elegant cascade that translates a single external signal into a massive intracellular response.
It is, which makes the pathophysiology of type 1 diabetes mellitus so tragic when you think about it.
Yeah, the sources describe type 1 as an autoimmune destruction of the pancreatic beta cells, heavily involving genetic susceptibility via HLA genes. The classic triad of symptoms is polydipsia, polyuria, and polyphagia, so excessive thirst, excessive urination, and relentless hunger.
Let's view those symptoms through the lens of everything we've discussed today. The beta cells are gone, so the patient has absolutely zero insulin.
Right, so after a meal, the patient's blood is incredibly thick with glucose.
Yeah.
But insulin is the mandatory key required to insert the GLUT4 transporters into the membranes of muscle and fat cells.
Without insulin, the glucose just bounces off the cells.
It's a profound paradox. The body is effectively starving to death while swimming in an ocean of nutrients.
And because the intracellular sensors detect starvation, the body panics. It releases massive amounts of the counter-regulatory hormone glucagon.
And the glucagon signals the liver and adipose tissue to aggressively break down stored fats.
Yep. The liver takes all those free fatty acids, runs them through beta oxidation, generates a massive surplus of acetyl-CoA, and converts it all into ketone bodies. Keto acids.
It's desperately trying to feed this starving brain.
Which brings our deep dive full circle right back to the operating environment from our very first segment.
Oh, the buffer system.
Yes.
The liver is dumping an overwhelming load of keto acids into the bloodstream. This is the massive heat wave in the server room.
Exactly.
The bicarbonate ions are rapidly consumed trying to buffer the acid. The lungs start hyperventilating Kussmaul respirations, desperately trying to blow off carbon dioxide to compensate. But the sheer volume of acid overwhelms the system.
The blood pH drops below 7.35. The patient plunges into diabetic ketoacidosis. If uncorrected, the low pH will begin denaturing functional proteins across the body. The biochemical software crashes, and the patient goes into a coma.
It is terrifying, but it proves that no pathway operates in isolation. An autoimmune defect in the pancreas rewires lipid metabolism, which overwhelms the blood buffer system, which forces the lungs to hyperventilate, which ultimately threatens the structural integrity of every protein in the body.
It's all connected, so to lock this material in, let's put you, the listener, on the spot with a clinical scenario derived directly from the text.
Okay, let's hear it.
Imagine you're evaluating a patient who comes to the hospital with excruciating colicky abdominal pain. They have a history of these attacks. The pain is so severe it mimics a surgical abdomen. They look like they have a ruptured appendix.
Wow.
In fact, these patients often have a history of negative exploratory laparotomy surgeries that found completely healthy organs.
Just unnecessary surgeries.
Exactly. But you remember a strange piece of medical trivia. You asked the nurse to leave a sample of the patient's urine in a clear cup by the window. A few hours later, exposed to the light, the urine has turned the dark color of red wine, the classic windowsill test.
Okay.
Based on the metabolic bottlenecks we've explored, what is the genetic disorder and what specific enzyme is deficient?
Take a second to think about it.
Got it. The diagnosis is acute intermittent porphyria. It's an autosomal dominant disorder characterized by a genetic deficiency in the enzyme uroporphyrinogen I synthase, which is part of the heme synthesis pathway.
That because of this bottleneck.
Porphobilinogen and delta-aminolevulinic acid accumulate to toxic levels. It's this toxic buildup that assaults the nervous system, causing the severe abdominal pain and neurological symptoms.
In the urine.
The porphobilinogen oxidizes in the sunlight to turn the urine that deep, dark red.
It's the ultimate lesson of biochemistry. You could open that patient up 50 times looking for a physical obstruction, and you would never find it. The obstruction is molecular.
Truly invisible to the naked eye.
As you go back to the wards or back to your studies, I encourage you to look past the hardware. The next time a patient presents with an isolated symptom, a cramp, a strange respiratory pattern, lethargy, ask yourself what invisible metabolic pathway is bottlenecking in the background. What is the systemic consequence of that single missing line of code? Keep pushing past the physical and keep looking deeper.